Say about Pieter. And in Amsterdam, he's the director of the Netherlands Institute for Neurosciences, and he recently started working at the Institute de Vision in Paris 1 day a week. Is that right?
Yeah.
So also, not be too bad a commute between Amsterdam and Paris. So, and one of the reasons that Pieter's in such high demand is he really covers the whole gamut of visual neuroscience, from monkey visual cortex chandelier cells to fMRI in humans, to his real core work in non-human primates, studying the physiology of visual cortex and how it contributes to visual perception. And I think, Pieter's done so many amazing studies. I'll tell us just about a few of them today, but if you haven't read his seminal papers on attention and perception, they're really worth reading. But really, what really stands out for me is sort of his intellectual frankness, I would say. When he was a grad student, he worked in Wolf Singer's lab, and Wolf is famous for originating the theory of binding through neuronal oscillations or synchrony. And this is, of course, the idea that we see an object in the visual world, and parts of our visual cortex are specialized for color and form or motion, and somehow we have to bind together those visual attributes so we can get a full picture of the objects that we're seeing. And the idea of binding by synchrony or neuronal oscillations is incredibly attractive, and it's still very popular, I would say, today. But when Wolf and Pieter designed these experiments, and when Pieter spent years doing these experiments, he had sort of the nightmare of every grad student, which is the experiments didn't work. They didn't support the amazing idea of his mentor. But Pieter was not discouraged by that, and he didn't try to massage the data or make it fit and somehow continue on in that vein. He just admitted, well, this data does not support that hypothesis, and now he's developed 20 years later a new idea for how neuronal firing rate increases could create assemblies of neurons and explain the binding problem that way. But I think it's really a great example for all of us that we should take what our data is telling us and fit our ideas and models to suit the data rather than the other way. So as an MD PhD, Pieter is perfectly placed to translate all these amazing discoveries he's made about the function of visual cortex into devices that can help human patients, especially patients with blindness. And today he's going to tell us about some of this work in his talk entitled Visual Perception and Visual Consciousness and the Restoration When the Eyes Fail.
So thanks a lot, Michael, for a very nice introduction. Yeah, it's true. When I got those results, so I just started to work in a new lab, and I wanted to test for once and for all, demonstrate that oscillations are doing binding, and it didn't work. So I actually thought it was the end of my career. So, and then I found this firing rate effect that I first thought was a nuisance, but it was actually the signal. So it's really a big pleasure to be here. I already had a wonderful morning, and I look forward to the afternoon. And so today I'm going to talk about the visual perception and a little bit about consciousness, and then work that we're doing in an attempt to restore vision in those people who lost their sight, quite in line with the work that Michael, Daniel, Yoshar, and other people here are doing here at the Institute. So I would like to, to disclose that we also started a company called Phoenix that aims to make this prosthesis a reality. And the reason that we did that is we realized that we have to go beyond the work that is typically expected from an academic. So if you really want to make something that is admissible in human patients, you have to do stuff that is outside my comfort zone, but still needs to happen. So this is the overview of what I'm going to talk about today. First, a brief introduction on feed-forward and feedback processing. A little bit related to binding, actually. Then I'll say, well, maybe the first 20 minutes or so talking about visual awareness. And then the large chunk of my talk is going to be about our efforts to create an interface with the visual brain of people who are blind. Okay, so feed-forward and feedback processing. So this is actually a slide about binding. So if you see a complicated picture like this one, your visual system is sort of confronted with a lot of information, colors, textures, you name it. So we don't have a hard time seeing that this head of the zebra is the same zebra as, for instance, this leg. Now that is maybe intuitive and seems immediate, but it's not something that the visual system gets for free. And the reason is that visual processing at early levels of the visual system starts with fragmentation. So neurons in low levels, retina, LGN, V1, you name it, they have small receptive fields, and these neurons, at first sight, seem only to care about what's going on in their tiny receptive fields. Okay, so vision starts with fragmentation, but this is not how we perceive. We perceive elongated, coherent objects. Now, when I started my lab in Amsterdam, I thought this is going to be solved by neuronal synchrony. It turned out not to be the case, not at all. But it's solved by a process that we call incremental grouping, where those image elements that belong together, that are part of the zebra that you're interested in, are labeled with an enhanced neuronal firing rate. And so this labeling process, psychologists call it object-based attention, happens actually for only a single object at any one time. So if the system sees a picture like this, your brain creates a labeled representation where the thing you're attending to, all its features are labeled with an enhanced response, and this generalizes to colors and other features, and thereby binds all these features into a single coherent representation. In my introduction, I cannot avoid showing this very famous slide by Velleman and Van Essen. I guess everybody knows it. This is also from the same authors, and it just illustrates the hierarchy, complex hierarchy of a visual system. You study it in non-human primates, also in humans. They're quite similar. So here's the retina, LGN, and then information arrives in primary visual cortex. And there's this wave of feed-forward processing, where information is sent from lower to higher areas, and low-level areas like primary visual cortex extract simple features such as the orientation of an edge. Neurons in intermediate levels of the system, like area V4, are tuned to feature constellations of intermediate complexity, and neurons at high levels are tuned to object category and other things. But there are also feedback connections here in blue and lateral connections, allowing for recirculation of activity. And this is actually the phase where we think that this incremental grouping takes place. So that probably relies on feedback and lateral connections. Okay, labeling these image elements that you care about. Okay, so much for my introduction. So I think we have the background now to tackle one of the more interesting questions in neuroscience. So what about awareness? What does a visual stimulus need to do to reach conscious awareness? Okay, I think that's quite an interesting question. I'm going to ask you to participate as my subjects. I'm going to present a stimulus around here very briefly. So please pay attention. If you see it, please raise your hands. If you don't see it, don't raise your hands. Okay, 1, 2, 3. Okay, that was easy. Let's start over again. Now I'm going to make it more difficult. 1, 2, 3. Who saw it? Nobody? Okay. Try again. 1, 2, 3. Yeah, I don't control the luminance of this monitor, so this can go wrong sometimes. Okay. In reality, there was nothing on the second, and my intention was to show something very dim on the third iteration, but it didn't work. So what happens sometimes, of course, that people raise their hands when there's no stimulus. It's nothing to be ashamed of. It's called a false alarm. And the nice thing is that for each and every one of us, there will be a contrast level. And actually on my computer, I can see this one, but on the display here, you only see this one, and this one is not visible at all. So you can imagine that for everyone, there is a contrast level where sometimes you see it and sometimes you don't. And that's very convenient because it means that you can show the very same stimulus to the same subject, and in some trials, this stimulus will cause awareness, and in some trials, it will not. And that's what we set out to do. And the situation that we obtained, and that was also very predictable, is very well described by the so-called signal detection theory, very famous theory proposed by psychologists many years back. And they proposed that if you present a weak stimulus, then across trials, you get the distribution of something they called internal signal strength. And so across trials, this distribution is shown here in green. So if I present a very weak stimulus, we take a sample from this distribution on the particular trial. And now in the signal detection theory, there's also a threshold. And if this internal representation crosses the threshold, is higher than the threshold, the subject will say, yes, I saw a stimulus. We call it a hit. But probably because of the stochasticity inside the brain, sometimes it may happen that I present the very same stimulus, but it stays below the threshold, and then the subject says, no, I didn't see it. So that's called the miss. Now, if there is no stimulus whatsoever, then we are sampling from another distribution with a lower mean. So in signal detection theory, it's typically centered on 0. And then typically, of course, the signal strength will stay below the threshold. Subject says no. It's called a correct rejection. But occasionally it may cross the threshold, and then the subject reports a stimulus that was not there. So that's a false alarm. And the questions that we asked here is, where is this internal representation of signal strength in the brain? What causes this variability across trials? So basically the width of these distributions. And what is this threshold? What does it correspond to in the brain? And when we obtained the data, actually it turned out that we started to support a theory on visual consciousness that was proposed by Bernard Baars and made very popular by Stan de Haan, so-called global neuronal workspace theory. That posits that if you present the stimulus to the eyes, it goes through the LGN, comes into the visual cortex, and gets propagated to frontal areas. And in the neuronal workspace theory, these frontal areas, they cause something that is known as ignition. And what is ignition? It means that maybe through recurrent interactions between neurons, this activity pattern becomes self-stabilized, so that if you take the stimulus away, it's no longer visible. These cells keep firing, persistent firing, as if they kind of carry a memory trace of the stimulus that was only briefly presented. And the idea then would be that on those trials where the stimulus is not consciously perceived, that this wave of propagation towards frontal cortex would be weaker and ignition would not occur. Right? So it's the ignition process that kind of determines whether something enters into conscious awareness. So we trained monkeys on this very same task you've been doing. So on 50% of trials, we presented the stimulus, very similar. 50% of trials we did not. And if the monkey saw the stimulus, he was supposed to make an eye movement to it after a delay. And there was always this dot on the screen. And the task of the monkey in case of no stimulus would be to make an eye movement to this so-called reject dot. And the animals were really good at this. So if the contrast was high enough, say larger than 10% of the stimulus, then they always saw it. But then, of course, if you decrease the contrast at some point, they started to find it difficult. And at some point, they had really a hard time seeing it. Okay, no surprises there. Now we put two arbitrary thresholds, one at 40% correct and one at 80% correct. And we said if the monkey's accuracy is better than 80% correct, the stimulus is really easy. It's intermediate if it's below 80 and 40%, and below 40%, that's a really difficult stimulus just to kind of analyze the data later. And then we recorded from area V1, primary visual cortex, area V4, mid-level visual area, and prefrontal cortex. And actually, we are recording from an area that is involved in the generation of eye movements. So that has an implication because I'm not going to dissociate the plan to make an eye movement from the conscious perception itself. Okay, this design is not allowed. So when we started to record, we were actually, or I was in for a surprise. Here you see the activity in primary visual cortex. In green, for those trials that were difficult, where the monkey detected it, it was a hit. And in red, where it was a miss. So somehow there's already variability in the propagation of information from the retina to primary visual cortex, it seems, because this happens already at the very first spikes that you see in primary visual cortex for a new stimulus. And we made sure in the analysis that the contrasts on the hit trials and mistrials was the same. Now, of course, if you increase the strength of the stimulus, so it's of intermediate difficulty or easy, then V1 activity increases. This is just higher contrast. But it's also remarkable that this difference between hits and misses was maintained at higher contrasts. And also that the easy mistrials, actually, that's this red peak here, elicited more activity than the difficult hit trials. But it has an implication. It means that if V1 neurons fire at a certain level, there's no guarantee that this stimulus will reach conscious awareness. Apparently, information can get lost downstream from V1 on its way, maybe to frontal cortex, where ignition might occur. In V4, we basically got the very same results. So here the difference might be even a bit more pronounced here. But also here the easy mistrials generated more activity than the difficult seam trials. Okay, so a certain level of activity in area V4, again, is no guarantee that the stimulus will reach conscious awareness. Now, in dorsolateral prefrontal cortex, the situation was categorically different. Whenever the animal was going to see the stimulus, were difficult or easy, we always got a high level of activity. And when there was no stimulus, or the monkey did not report a stimulus, activity was always low. You see that on those trials that were easy, there is sort of a ramping of activity that somehow doesn't reach enough activity in frontal cortex. Okay, so here you actually could put the thresholds, and if the activity crosses the threshold, maybe it becomes self-sustained. And we also looked at those trials without the stimulus in the receptive field, and in black are the trials where the monkey correctly rejected, so reported no stimulus, and in blue the trials where he committed the false alarm. And you see that in dorsolateral prefrontal cortex, activity reaches the same level as on hit trials. Probably we're just reading out the eye movement plan here. You also see that there's an impact. This is significant. It may seem subtle to you, but it's significant. You see actually a slightly higher firing rate in V1 and V4 on the false alarm trials. Now these slow ramps, they are famous in neurophysiology, and actually I strongly believe that this is an artifact of averaging across trials. So I think underneath, there might be moments in time where the monkey suddenly kind of has ignition and starts to imagine that there was a stimulus there, but I can't prove that. It's very difficult to prove that. Because if there are many trials with fast ramps, but these ramps happen in different trials at different time points, then if you do the averaging, you get the slow ramp. Again, I can't prove that. So of all these areas, there's only one that we recorded from that sort of is compatible with this internal representation of signal strength, and that is clearly dorsolateral prefrontal cortex, with most activity for hits and false alarms, and least activity for the misses and the correct rejections. Okay, so when I talked to Stan de Haan about this many years back, he said, oh, that's really cool. He got really excited, and he had published also models with very similar predictions. And then he said, you know what I'm going to do? I'm going to make you a model that is as simple as possible that can account for all of your data. And so this is his model. And his model of the data consisted of a total of 5 neurons, 5 stochastic neurons. So he had 1 neuron in the LGN, 1 in V1, 1 in V4, 1 in parietal cortex, and 1 in frontal cortex. And you see that there are feed-forward connections, feedback connections, and self-connections. So if you then present the stimulus, it propagates through the network, reaching frontal cortex. Now, in the model, he made the self-connections and the connection between parietal and frontal cortex a bit stronger, so that if activity reaches a certain level there, then it can self-sustain. And if that happens, if you then take the stimulus away, activity will stay in the network, and well, you can then think that the network will report that it's all stimulus. Okay, that would be a hit trial, and then because of these neurons are all stochastic, can present the very same stimulus, but it may fail to cause ignition, and then if you take the stimulus away, activity will collapse in the network. Okay, and it would be a mistrial. So this is activity of the model neuron in V1, and you see that it reproduces many of the results. For instance, that the easy mistrials give more activity than the difficult hit trials. Also in the model, activity can get lost downstream from V1. V4 is basically the same. And in dorsolateral prefrontal cortex or frontal cortex in the model, there is this possibility to self-sustain activity. Therefore, you always get this ramp and it reaches a certain asymptotic level, always. You also see that on the mistrials with a strong stimulus, there is a bit of ramping, but it apparently is not enough to reach self-sustained activity. Now, on the false alarm trials, we also see those ramps, but in the model, we can be really sure. In the model, we see these fast ramps that happen at different time points in different trials. Okay, so this is the model. Kingston made it on a rainy Sunday afternoon. This is our data. This is the hard work of 3 PhD students in the lab, working for 4 years to collect the data. So if you're still thinking about career choices, of course, data is also very valuable. Okay, so conclusions. So I think this suggests that the variation in internal signal strength across trials are probably explained by variations in the efficacy of this feedforward propagation from the retina all the way to frontal cortex. The threshold for perception is the threshold for this ignition, this production of self-sustained activity, working memory it is. And false alarms in the model are explained by spontaneous ignitions. They are very rare, very seldom, but sometimes it happens that just spontaneously the activity crosses the thresholds for self-sustained activity, even if there's no stimulus. So based on these results, we might now be inclined to think that most of the interesting machinery for conscious awareness is in frontal cortex. And that is what some people in this consciousness debate like to claim. Well, I'm not in that camp, actually. I think they are important, these neurons there. But there are actually situations where you also need the visual cortex for conscious awareness.
I want to ask, why did Stan include the parietal cortex? And what's the reason? What's the role that plays? It wasn't in the external data you offered.
No.
And it's not a specific testable.
Not true. So I think you could, not necessarily. I think you could make the same model of skipping parietal cortex. You just make the self connection in frontal cortex a bit stronger to compensate for it. So I think the essence, but I mean, that would remain to be modeled. We haven't known.
So the idea is that there's more self-excitation.
Yeah, so because I think we know from many studies, including yours, there is a lot of persistent activity also in parietal cortex. So that's why it makes sense, but it's not essential for the moment.
Do you know what needs to be achieved when there's population without inhibition? What needs to be achieved?
If it's a mistrial?
In any case.
Oh, so if there's persistent activity when there's working memory activity, what terminates it? Yeah. So in this model, probably would be hard to stop the self-excitation. Yeah, there must be very powerful mechanisms in the brain that cause forgetting when it's necessary. So you have these attractor-like structures that support a working memory, but if the task changes, you should just abolish the working memory and focus on another working memory. So that's, I think it's a study in itself, probably many studies in itself to answer that question, but I think it's very important to have that.
So it sounds like you're defining, so we define awareness as sort of the time at which you get this sort of attractive dynamic. So it's this nonlinear and this dynamic process.
Yeah.
It's sort of a switch. So I can ask this right. So I mean, so it sounds like it's almost like you know, it's accumulation of bound and cost of bound and then
Yeah.
Right. How do you think about this in terms of variable bounds, right? I mean, just like you started with the mapping of similar attention theory, concept awareness is always really weird to me when you're trying to distinguish the decision variable from the threshold, right? This seems like you're defining it as a binary threshold The point of signal detection theory is that there's a graded
Yeah.
representation underlying it, and it's a separate process that governs when you report it.
Yeah, so in this very simple model, these two phases, so to say, are separated in time. Right, so there's the phase that the evidence comes in, and then the attractor dynamics kick in, and it's kind of converging to one or the other state. But hold your breath. I'm going to kind of dilute this story. I don't think that's the whole story at all. So I'm basically on your side. It's just for a very simple weak contrast detection task, it does a reasonable job in explaining the data. That's it. Yeah. Oh, here before you mean? I think because it's a sorting exercise. So you're sorting trials where there was no stimulus. And so if there is already some lingering activity in the system before the stimulus appears, there's a high probability that it will kind of cross the boundary for self-sustained activity. Actually, we saw the same in the monkey data. So when we started trials in the monkey data with false alarms are actually a higher propensity to generate a false alarm, then we also saw a higher level of ongoing activities, sort of spontaneous activity in V1, in V4, and also in dorsolateral prefrontal cortex. They're just closer to the bound, so the probability of false alarm is just higher.
Is that like a failure to normalize to the baseline?
No, it's a real signal. It's real. It's really there. Yeah. Okay, now let me then start diluting the story, because I don't think awareness is only in frontal cortex. So what I'm going to do, and with Zoom, this might not work so well, so we'll see how well this works. I'm going to very briefly present to you a visual stimulus, very briefly, and it's a complex stimulus. So try to see what is there. Okay, 1, 2, 3. Who saw something? Whatever, colors, shout. Go ahead. You saw a bird. Everybody saw a bird? Or did you see wood chips, textures?
Yeah, perfect.
Okay. Anything else? So I think most people, you're the exception. This is what you saw very briefly, and there was a bird. But it's hard to see, right? So here you see the bird. So this really requires figure ground segregation. And that typically takes a bit of time. And this is actually where I strongly believe that you also rely for your conscious perception on interactions, maybe between frontal cortex, but definitely also with contribution of visual cortex. And this here is basically where you need this labeling process that I was talking about to occur, to do the segregation. Okay, so this is a very simple variant of the same problem. So most of us will see a square with a certain orientation superimposed on the background with a different orientation. In fact, it's just a bunch of line elements, and a visual system creates the square perception. And my good friend and colleague, Victor Lammers, started to study that many years back. And he already back in 95 demonstrated that neurons in primary visual cortex care about the difference between a figure and a background. So what you see here is a small receptive field on a figure, here the same receptive field, and if you look carefully, it's the same image elements in the receptive fields on the background. And what happens here is that neurons care about the difference, so they have extra activity if their receptive field is on a figure. First, you get the feed-forward response. That is the same because what is in the receptive field is the same. This is basically when the information comes from the eye in primary visual cortex. Then V1 sends the information to higher areas. It starts to feed back. And then in this recurrent phase, the V1 neurons that have their receptive field on a figure start to enhance their response. Now, in a very nice experiment, Victor and his colleagues also demonstrated that this difference response, we call it figure ground modulation, is there only on those trials in which the animal perceives the figure ground perception. So here in this situation, you see that the feed-forward response is the same. So probably the monkey always saw there's texture on the screen, but on a small percentage of tasks, he failed to see the figure ground perception. And then his figure ground modulation was also gone. Now, what we wanted to do, and that has been a holy grail in the lab, is to do a causal manipulation, really knock out this figure ground modulation and see what happens to perception. But that turned out to be very difficult. So we started to work on optogenetics in monkeys. We found it hard. So here you see the figure ground modulation in V1 of monkeys. Actually, it's a signal that generalizes quite well across species. So here we recorded from area V3 of humans. So these were humans with epilepsy, and we have a collaboration with neurosurgeons in Amsterdam who then have these so-called SEG electrodes that they put into the brain to localize the source for the epilepsy, and we have ethical permission to use these micro wires that extend from the tip of the electrode so that we can also record some single and multi-unit activity. And this is spiking activity in V3, and you see that also in V3 figures elicit more activity than the background. Now, for the purposes of causal manipulation, it's nice to see that this also happens in mice. Okay, so also in mice, figures elicit more activity than the backgrounds. So now we went to mice, and we spent a lot of time trying to train mice to discriminate between the figure and the background. It was not super easy, but in the end we worked it out. So here's a figure ground stimulus, and the mouse licks to the left of a double-sided lick spout if the figure is on the left, and he licks to the right when the figure is on the right. Then we used optogenetics. And so if this is the figure ground response, we used a very powerful optogenetic silencer. It's called GTACR2. We can wipe out all the spikes from primary visual cortex. It's really powerful. Now that's interesting, but of course now we are also wiping out the feed-forward response. Now the nice thing about optogenetics is that you can let through the feed-forward response and selectively knock out the late spikes. So what will happen to perception? And you can let through more and more of this delayed response as well when you see the figure ground modulation. And this is work that was carried out by Lisa Kierberger in the lab. We actually also trained mice on a task that just requires them to detect contrast stimulus on the left or contrast stimulus on the right without the need to do figure ground. Now what happens if we knock out all the spikes? Well, actually the mouse is largely blind. He fails in both tasks. He's slightly above, some mice are slightly above chance level in contrast perception task. Maybe because there's also some pathway through superior colliculus or so, we don't know. But on figure ground, they're really at chance level. Now, what happens if we let through the feed-forward response, but knock down the late spikes in V1? Then the mice are perfect on contrast perception, but completely at chance level at figure ground. Okay, we really have to let through some of this late V1 activity that is modulated by this figure ground perception. And only then the mouse can do the figure ground task. Okay, so we are really excited about this result because it suggests that this late phase of the V1 response is important for the perception of a figure ground stimulus. Okay, so I now believe actually that what's going on in the back of the brain is sometimes, certainly for this more difficult perceptual discrimination, is really important for conscious visual perception. And so if you think about the global neuronal workspace, in some situations, it might be useful to include neurons in maybe even up to the level of primary visual cortex into this global neuronal workspace. Okay, so short conclusion. So there are aspects of conscious perception that rely on interactions between V1 and higher cortical areas, probably including frontal cortex. And now we'd like to switch gears a little bit. Of course, this is a whole set of fundamental knowledge that many labs across the world gathered about visual perception. So we are now going to exploit that in the idea to create an interface with the brain of people who are blind. And the reason that we got into this is that we use these youth arrays, and it became a habit in our lab to implant at least 200 electrodes. And then we thought, let's ramp this up. And there was a very talented postdoc in the lab, Sing Chen, who came and we said, let's try to do 1,000. So that's what we set out to do. I think this narrative is very familiar to you, given the work of Michael, Denise, and Daniel, and more people here. So in the world, there are 40 million people who are blind, and it would be great if we could recreate a rudimentary form of perception. Now, we are now looking at the primary visual cortex, but I'll also say a few words about the LGM later. And we know from the work of many labs, started with Gals Brindley in the 60s, that if you place an electrode on or in primary visual cortex, you do a little bit of electrical stimulation, then a person can also be a person who has been blind for quite some time. You'll see a dot of light at the location in the retinotopic map where you have your electrodes. Now, if you would have 1,000 electrodes or more, you can create 1,000 of these dots of lights at different positions. And of course, then you can try to write by switching some of these phosphines, as we call them, on and leaving some of them off. So a future system, and many people have proposed this. This is definitely not unique to what we are doing in our lab. Propose that subject would have a camera, could be mounted on glasses. You can just buy those. And you can use maybe AI algorithms to simplify. And actually here we're using what we know about perceptual organization and figure ground. One idea would be to place the phosphines on the contours of the objects that matter for your task here as to navigate. So you can actually, based on artificial intelligence, leave out a lot of the clutter that is not informative. Now, this is probably not a super easy problem, but it's not a problem that we are really too concerned about. Then you need to send it basically to the brain, and then you need the brain chip and also electrodes in the brain that are going to do the stimulation. That's the part that to me seems hardest. And so what we did, and this is the work of Singh, I already mentioned her, is we implanted 16 of these so-called Juta arrays in the visual cortex. So each array in our case had 64 electrodes, 8 rows and 8 columns of electrodes made of silicon. This is what the implant looks like. So this is a percutaneous connector. So this is visible outside the skin. This stuff is screwed onto the skull of the monkey. These are bundles of gold cables, 16 of them, with at the end these Juta array. And these gold cables, they're basically running on the skull. At some point they dive down below the skull, and then these electrode arrays, they're punched into the primary visual cortex. And this is how we implanted those. So 14 of them went into primary visual cortex, 2 of them in the area before. And then we mapped out the receptive fields of the neurons that can also record. We can not only stimulate, but also record. And I'm just going to show you in a moment how we map these receptive fields, but I guess most of you will know how this works. These monkeys were not blind. And what you see here is a very nice representation of the retinotopy, right? So this is one lateral array. You see the receptive fields are close to central vision. This is closer to the midline, and then you get receptive fields that are at a more eccentric location. And we replicated the result in the second monkey. So here, this is a movie. I think we get it to run. So here's the monkey looking at the screen. Of course, you would not see this in the lab. This is what Singh made as a mock-up. You see the arrays, and she plotted yellow on those electrodes with a high level of activity. So basically what you see is then when this bar of light goes through the receptive field, it touches one receptive field and then another one and then another one. And that results in this wave of activity in primary visual cortex. Now you can also plot these waves in the visual field. So now I'm going to show you the activity at the location of the receptive field. It's basically the same data, but plotted differently. So the red dot is the fixation point. And you see that the bar of light is trailed by a wave of activity. And the delay between the bar and the peak of the wave is basically the delay between a cell in the retina and a cell in primary visual cortex. This is actually slowed down by a factor of 5. Okay. Now, we are of course not so interested in recording. We now know where all the receptor fields are, so now we can start stimulating. Started by stimulating one electrode at any one time. I'm going to show you the receptor fields that we're stimulating here in white. And the monkey's task is to make an eye movement where he perceives something. So you see he makes an eye movement approximately to the location of the receptor field of the cells we're stimulating. You may also notice there's a bit of a deviation here, but I think that's just the calibration of our eye camera. Okay, so this has been shown by other labs before us. So it was interesting that we could do this for hundreds of electrodes. But the big question for us was, can we also generate pattern perception? Can we impose meaningful patterns onto the primary visual cortex? So to test this, we trained the animals to recognize letters. So here we present the letter D, then there's a choice menu, and the task of the monkey is to make an eye movement to the letter we just saw. And then in the exciting part of the experiment, we are not showing letters anymore, but we are kind of imposing a letter shape by stimulating cells, and we know where their receptive fields are, just trying to paint a letter, either letter A onto primary visual cortex. And actually, just before we published this, Michael, Denise, and Daniel showed already that this is possible also with surface electrodes. With the surface electrodes, you cannot stimulate everything at the same time. You have to do sequentially, because with the surface electrodes, you have to give milliamps, and if you then try to stimulate 20 electrodes at the same time, it would just be too much current. So these are electrodes in the cortex, and we can stimulate with microamps. So we can do this. And this shows you the results. So we're stimulating a set of electrodes in the shape of a letter L. And yes, the monkey makes an eye movement to the appropriate letter. So we thought it was so cool. We made a dance in the lab and we got the result. Okay, so usually the monkey is right. He makes an occasional mistake, but he can do this. So this is a demonstration that you can indeed write meaningful patterns to the brain. I think now he's going to get it wrong. Yeah, it's real data. Okay, so this is then where we now stand. And so, as I said, I think this part of the scheme is probably solvable, but I'm not claiming that we solved the other parts. And we are now working together with a researcher in Spain, Eduardo Fernandez, who also implanted the UTA array in primary visual cortex of a blind person. This is Bernadetta Gomez. She's now actually quite famous in Spain. And this is where he implanted the electrode. It was in V1. And here he stimulates a set of electrodes like we did in the monkeys in the form of a large square. And Berna reported seeing a large letter O. Here, Eduardo and his team stimulated these 4 electrodes, and she reported seeing a small letter O. So that is great, right? But it was not always as straightforward. So here, Berna reported seeing a letter I, which I can still kind of live with. But here, she actually reported seeing a letter L, and I don't see the L. Okay, so what's going on here? So you have to realize that in Berna, there's only one array that's 4 by 4 millimeters. So I think what we're seeing here might be, but of course I can't prove it, but it might be the reflection of uncontrolled activity, some interference patterns that happen between the electrodes that we're actually stimulating. I can't prove it. Now in the monkeys, of course, the electrodes were spread across 2 centimeters. So these degree of interference must have been much lower in that situation. And here is Berna. Now this is the thing that I was trying to set up. I hope it works. is now, for me, Bernadetta is really a hero because she really realized that this was an experiment. It was not a treatment, right? So also her electrode array needed to be explanted after 6 months. So she undergoes 2 times brain surgery in a self-visualist way because she says, I want to do this research because I want to benefit future people who might get a treatment, realizing that this was not a treatment. Okay, so but and that is where we need more experiments because there are several problems to be solved. And these are, for me, the 2 kind of most urgent problems. That's the coverage of the visual field. So with 1 electrode, so I'll show you. These are in the visual field, basically where Berna saw her dots of light, or phosphines. And so this is a very tiny region of the visual field. Actually, if this is the visual field, this is where Eduardo and his team could produce phosphines, right? So it's less than 1%. So it's not useful. And another problem that we have is longevity. So of course, we went to the extreme in planting UTA arrays, and you should realize these are just tiling UTA arrays in primary cortex, gluing them all together, gluing them also to the cortex. Then there are these 16 bundles of gold wires that become something that's quite fixed. So when the monkey does this, then you might imagine that quickly be a bit of damage. Actually, we were able to get good stimulation results and recordings for a year, but after that, it quickly started to deteriorate. And this shows you why. So basically, what you see here is a huge lesion. Right? Basically, we punched a hole in the cortex. And what you see here is now we are doing a sort of perimetry task in which we present small dots of light at different locations in the visual field. Now, when it's yellow, the animal can do it very well. And the black contour is that collected the receptive fields of the electrodes that we had implanted. And you see that the monkey's vision at those locations actually became quite poor. Not outside where we implant the electrodes. So this seems to be really the result of our procedure. So this is not a treatment, right? So we need to solve this. And this shows you also after explantation what these electrodes look like. So you see that these electrodes, these needles have been encapsulated by glial tissue. So is there hope? I think there is. So we are working together with labs, including the one of Zhongxia, that make these highly flexible electrodes, or actually shanks you should call them, with electrodes on top of them, from, for instance, polyimides. So you can make them from these polymers, and you can, in the clean room, pattern the metal on them, and they can be very thin, up to 1 micron thick. So they become very, very flexible, and they seem to float basically in between the neurons and cause very little damage. So that's the direction we're thinking in. And to solve the longevity problem, actually, in our lab, we are now considering actually going away from primary visual cortex because it has this, on the left side, 25 square centimeters of tissue, and on the right side, 25 square centimeters of tissue. Part of it is, a large part of it is actually buried in sulcite, so it's very difficult to reach. So we're actually thinking of the LGN. Now, we're not the first to do that, so there's actually very beautiful work from Jon Pesaris and Clay Reed many years back, demonstrating that in the LGN you can also create phosphines. And we're actually now making a new probe that's going to place these flexible electrodes in the LGN. That's the idea. And what you see here is the probe. We developed it here for mice. And what you have here is a flexible electrode that is actually bound to a tungsten wire. You put it into the LDN and then you retract the wire, and the flexible shank, so to say, stays in the tissue. And here, this shows you some micro CT images of successful implantation in the LGN of a mouse. And here we just show checkerboard patterns to demonstrate that it indeed was in the LGN and not in some random place in the brain. It's good. So we are now actually gearing up to try this in monkeys. And so if this is work from Chris Klink, who simulated, this is not real data, it's a simulation. We know the retinopathy of the LGN that has been published. So where would then the phosphines end up in the visual field? And that is shown here. Right, if you place electrodes on one LGN, you might actually get quite a decent coverage, especially of the central 20, or what is it? Central 10 degrees of the visual field. Of course, you still miss out on some of the retinopathy there. But it's much better than I think is achievable by interfacing with primary visual cortex. Now, can you do that? Can you really go to a human and place an electrode like that in the LGN? Well, it turns out to be yes. So I came into contact with a neurosurgeon, Osvaldo Vilela in Brazil, who recently put a DBS electrode in the LGN. And he does it to treat epilepsy. So he treated 2 people with epilepsy, and he has good evidence that it originated from the visual cortex. And it turns out that he gets the epilepsy under control by doing DBS of the LGN, very surprising. Now, here you see the patient. Patient is awake during the whole procedure, fixed with his head. And this is, of course, what you normally would not see in the OR. So this is us. And so the patient is not blind, right? So we can do interesting visual experiments, which is what we did. So I'm not sure how well this will come through. So before the neurosurgeon places the DBS electrode, there's this mapping procedure with a high impedance electrode so that you can really record from single neurons. So this is single neurons from the LGN. And you see the neurosurgeon flashing a light. Do you hear the entrainment? I think maybe only the EFIS aficionados will appreciate this. Okay, so we can a neurosurgeon can place an electrode in the LGN. It's not a place that is impossible to reach. Here you see actually a post-operative MRI, and you think, wow, that's a big lesion. No, that's an artifact. So there's not a lot of damage. You see these things also with normal DBS. And basically during the surgery, while the mapping was taking place with the high impedance electrodes, we did some very basic receptive field mapping experiments in the LGN of this patient. And here you see basically the response. So this is now eccentricity. So the bar moves from 10 degrees to 25 degrees. That's what you see here. You get a nice response, and it moves back from 25 degrees back to 10 degrees. You basically get almost a replica of the response. And here up and down. Now based on these data, you can then, here's the center of the visual field. You see that we mapped out an LGN receptive field. And you can do more interesting stuff. So here we use checkerboard stimuli that were phase reversing. And here the subject closed the left eye. So this was seeing through the right eye. And basically we saw no response, only some spontaneous firing. But when he closed the left eye, then we got decent response. And for some reason, the cell preferred one of the checkerboards over the other one. And so this is what you should expect from the LGN, of course, because there's layers that are connected to one eye and layers that are connected to the other eye. So I'm not claiming that we're anywhere close to a viable prosthesis for blind people, but I think this could be a promising avenue that actually was initiated by John Pesaris many years back. And we think that this is something that might in the future result in something that is of use to blind people. Now, the vision that they're going to get is not our vision, not normal vision. And you can simulate it in virtual reality, which is what you see here. Can you recognize what's going on? I guess you might. Right, so I think this is maybe an overestimation of the quality of vision through such a prosthesis. So it's nowhere near normal vision. Okay, so with that, I would like to close, but not without thanking the people who did the work. So I started to talk about consciousness. So Bram, Bruno, and Dev, they were the 3 PhD students who worked on this project for 4 years. These are the theoreticians, including Stan, De Heen. These are the people who did the work on the figure ground and on the mice. And here is the team working on the prosthesis projects and also on the LGN recordings with Osvaldo Vilela and Sergio Neunschon from Brazil, helping us or actually carrying out the surgery in the LGN. Thanks for your attention. John.
So, this, foray into the LGN is driven by the coverage issue. And, I wanted to ask, if, what role does the class system plays in this? As well? Because the coverage issue may not be as important if you could learn to resolve more information from the piece of tissue that is being, if it's like, figure it out.
Yeah,
So what's your thinking on that? Was there any learning in the monkey study was the origination of the question, but the larger question is.
yes, so of course, I think the jury is out, but indications are that that type of plasticity is not so overwhelming. Why do I say that? So there have been several attempts to kind of recreate sensory surfaces, so connecting camera, say, to something on the tongue or something on the back, skin, and these attempts were actually always abandoned by the subjects, the blind subjects who were supposed to benefit from them. Then you have there's a device called the voice that makes a line scan like this, and whenever there's something at a certain height, sort of a interesting item, then it gives, if it's high, it gives a high pitch, if it's low, it gives a low pitch. So basically by listening to the soundscape, so to say, you could work out. Was never successful. I think people have also looked in VR with simulated prosthetic vision, how much visual field coverage is useful to do everyday tasks. Also there, I think the consensus is that you need some minimal degree of coverage to be of use. But of course, there I'm not sure whether they really pushed it. So there you might want to push it, having somebody living in virtual reality for maybe 2 months or something, and maybe you then get the plasticity you want. But so far, it seems that people are maybe also wired up during their childhood to kind of, be familiarized with the sort of full visual field, and when you start chipping away from it, the functionality becomes less, it seems.
Great, well, looks like our speaker is on.